Hello?
Okay, so it's a pleasure to have Daniel Schultz here with us today. He's going to talk about gene regulation and antibiotic responses. So thank you for coming.
Thank you. Thanks for having me. So I'm going to try to convince you that antibiotic responses are like a very neat little system to study these issues of dynamics of gene regulation. Okay, so just like a broader picture, I'm interested in I'm interested in like how does a network of genes respond to a signal, and not really in terms of like what genes get turned on or off, but what happens right as they sense the signal. So say if in the case of antibiotic response, if the signal is at the presence of antibiotics, then what happens right as this as you add the antibiotic to the colony? So what happens to the expression of the genes? So usually a lot of people just think of this in terms of like what genes get turned on or off, but I'm going to worry about this part here, the transient, right after the drugs are added. And I'm going to try to convince you that this is actually a matter of life and death to individual cells, like if you look at this in the microscope. So I'm going to ask, like, what are the design principles that guide this dynamical behavior of the cell responses, and how can this process be optimized like for this transient? So to study this, we need not only to know the topology of the network, so the connections between the genes, but also we need to worry about the molecular details of this interaction. So say the binding and unbinding of these regulatory proteins to their binding sites, the production and degradation of molecules and things like that. And then we worry about like how is this whole process actually coded as a sequence in the DNA, and how does it evolve, and how does it evolve to optimize this whole process. So I'm going to try to make the case here that gene regulation actually evolves to optimize the dynamics of gene expression during the responses. So these are the kind of tools that we've used to look at this. So we do mathematical modeling, both using stochastic models, which I'm not going to talk too much about today, and also dynamical systems. We've done very careful measurements of gene expression during these responses, both at the population level and at single cell level using these microfluidic devices. They're pretty cool. And we look at evolution both doing experiments where you can propagate cultures in these little vessels here, and looking at genome databases that has like all the natural variation of this kind of mechanisms. So this is kind of the outline. I'm going to start more of like an introduction with citing some previous work that we've done and cell-fate decision in Bacillus. And then we're going to jump into antibiotic resistance. And then at the end, time permitting, I'm going to take a risk here and mention some more recent things that we've been doing, kind of a work in progress. Okay, so cell-fate decision in Bacillus. So Bacillus is a soil bacteria. So they're pretty interesting. So usually if you give them a lot of food, then they're going to take advantage of that, and they're going to grow as fast as they can. But when they start to run out of food, then they do many different interesting things. So this particular species, they can adopt like 10 different phenotypes, or they try to do different things. So they can develop motility and chemotaxis and try to swim after food. They can secrete some some enzymes to try to digest other stuff that they can find around, among other things. And what's interesting is that if you look at a population that's isogenic, like all the cells have the same DNA there, but you can see many of these different phenotypes coexisting in the same population. And here I'm going to talk about these two different phenotypes that this species adopts kind of like at the end of their life cycle. And this is more or less the network of genes that makes the decision of going into either of these phenotypes. So eventually the cells form a spore. So if they really run out of food, then there's no more opportunity to grow. So they form this spore that is very resilient, and they can just wait for thousands of years maybe until conditions improve. And in the process of right before sporulation, some of the cells in the colony, they go into this other phenotype of competence where they're able to import DNA from the medium. And they do that, it's believed that they do that not only to use it for food and for their own replication, but also to increase genetic diversity in the population. So here is a picture of a colony, of a starved bacillus colony. All the cells descend from the same cell, so they basically their DNA is the same. And he said a lot of them went into sporulation already, so these are spores. And a few cells in the population, they go into this alternative phenotype of competence, and they're competent for a while, and then they come back to vegetation. So, and what got us interested in this in the first place is because it's a stochastic decision, ultimately. And we wanted to look at this, the role of noise in cell fate decisions. So I'm going to start looking at a single gene and then a genetic motif, and then we're going to look at we're going to go to the larger scales to look at the decision at a broader level. Okay, so first we're going to look at just the transcription factor that controls entrance into sporulation, into competence. So it has this positive feedback loop here. And we're going to write this rate equation that, yeah. Uh-huh.
The word competence. What is competence?
So competence is the name of this phenotype where the cell produces all this machinery to import DNA from the medium. So the cell decides to go into this competence state, and then it expresses all the machinery that it needs to pull in DNA that's like swimming around in the environment.
One of the things that can happen if you starve the cell.
Yes, so if you starve the colony, a few of the cells, they go into the state, they stay competent for a while, and then they come back.
And this DNA is incorporated, or it just sits there in the cell?
So it's pretty interesting actually. So there's so many different species do this kind of thing, and there are some species that they try to recognize their own DNA, so they have means to recognize that the DNA is from their own species, and then they pull those in. And then there are cells that don't do that, but they have these quorum sensing mechanisms that are tied to the decision to go into competence. So they only go into competence when they know that all the neighboring cells are from their own species. So they do try to pull in DNA that's related to their own. So that's why people think it's like not only for food to be digested, but also to increase diversity. Yeah.
I didn't understand. It incorporates parts of external DNA, or it utilizes the amino acids?
So because they try to incorporate DNA that's from their own species, that it's kind of thought that this is to that they incorporate it basically. To increase the genetic diversity and also use for their own purpose of trying to replicate a few more rounds. So the thing is that this colony is starving, right? So there's a lot of cells dying, and there's DNA being released in the environment. So that's the point of it. But this is a subject of debate, even with microbiologists. Okay. All right. So we're going to write a rate equation here to describe the concentration of this transcription factor. So this transcription factor, when it goes to high levels, that's when the cell goes into complex. And so we write this equation, then there's like a few there's terms of synthesis and degradation of so N is the concentration of COMK, of this transcription factor here. Okay.
That's within one cell.
That's the concentration of COMK inside one cell. Yeah. Okay. So then there's a basal rate of expression here that we're going to consider, and then there's a activated rate of expression that's much larger than this one, that happens when the protein that's produced by this gene binds to the site here where transcription is initiated, and stimulates the transcription of this gene, right? So basically, this activated rate of expression here is modulated by the occupancy of this binding site over here. So that's this over here. So this is an expression that you get from the binding equilibrium here of the protein to the DNA. So and this is a degradation term. That's so this protein is actively degraded by this degradation complex over here. So say at small n, low concentrations, then this is close to zero, and the system is kept at a low concentration range, right?
Right, there's a critical concentration where the cell goes into competence phenotype.
Yes. So when this expression of this gene is very high, then the cell goes into competence.
That's not any C, something else.
So C is an exponent here that has to do with the cooperativity in this binding. So it's basically the degree of multimerization of the protein as it binds over here.
I don't understand something.
Mm-hmm.
First, as I'm seeing, you are talking about the transcription of this gene.
Right.
So N is an mRNA expression.
So I'm not considering that step. I'm going straight to protein level.
So you are talking only about protein level.
Only protein level. So this is concentration of the protein. Okay. Okay. Yes. It's a power. Yes. It's not a needle. It's a power. And it has to do with if this protein is roughly represents the multimerization. So some proteins bind as dimer, some as tetramer. Then that corresponds more or less to this power here. Okay. So then, if there's so let's say if the concentration is low and this is close to zero, so the protein concentration is kept at low levels, but there's noise in the expression. And if the concentration crosses this threshold and zero here, and this term and this here gets past half, then this starts to be relevant, and then then that increases the expression level a lot, and then the cell goes to this other stable state where now expression is very high, and you go to this to the high levels. So if we borrow this, make an analogy to this trick from physics and consider this as like a one-dimensional movement of an overdamped particle, and we think of this as like an external force acting on the particle. Right? Then we can integrate this force, and then we get to this known picture here of an energy barrier problem, basically.
Sorry, I got 6 points for this equation. Is it stable?
Yeah, so there's 2 stable points. So there's 2 stable points. So there's one at low concentrations and one at high concentrations. Right?
But why only 2? That depends on the C
So it depends in C in the sense that I think for C2 or higher, you need some nonlinearity there. So C2 or higher, you already get it. But there's two stable points.
There's no noise,
Huh?
At this point, there's no noise.
At this point, there's no noise. But I'm going to consider noise here. So say if there is noise in the expression here, then the probability they're going to transition from the low concentration point to the high concentration point is going to depend on the height of the barrier and the noise in the expression. Right? So that's so then you can actually kind of like calculate the probability of transition into competence. And then what the cell does that is pretty neat is that it can control the height of the barrier basically. And in this case, it does this with this by making this peptide come S here, which competes for degradation. And so that would be the noise. That would be the noise. But I didn't really explicitly put it in the equation. I didn't put it in the equation, but that would be the noise. And try to keep it a bit more simple. Anyway, so then we can write. Now let's consider this other protein here. It's actually a peptide explicitly, and we can write these types of equation for the expression of both of these, and consider this like a two-dimensional system. So what we do with this is so now we have a system of two equations, and we can go to this phase diagram here that has the concentration of both of these proteins. And we can plot these curves of the zero derivative here of both of these equations. And then where they cross, those are where both derivatives are zero. Those are the fixed points of the system in this case. So there's two fixed points and a saddle point over there. And depending on the parameters that you use, so there's a lot of work on this specific system that we found out that it's actually an excitable system where you have a stable fixed point at low COMK concentrations, and the system can get kicked out of there. Oops. Can get kicked out of there by noise, and it goes into these excursions around this second unstable fixed point, which kind of corresponds to what you see in the bacillus cell, where the cell goes into competence for about 20 hours and then comes back from that. Right? I'm not going to talk a lot about it today, but we did some work where we actually described the system not as rate equations, but we have a stochastic description where we consider the concentration of cells as discrete. So, the concentration of proteins as discrete quantities, and then we describe this system with a master equation where we have all the discrete states of the system and all the transition rates between all of the discrete states, and this is nice because it incorporates into the description all the noise and discreteness. And these kind of equations are hard to deal with analytically, but you can simulate them.
The transition rates are, how do you determine the transition?
So in this case, we would consider a production of protein or a degradation of protein or a binding of protein to the DNA. Those are all stochastic events where you know the rates, right? You know the rates? Where you would use the rates of those as a parameter. Yeah. Exactly. Yes. Yeah. Huh?
What exactly are you testing? How can you ever get the wrong answer?
Well, there are not that many parameters to fit, right? There's synthesis and degradation of proteins, and there's binding and unbinding to the DNA, in this case, and you more or less know what's reasonable from experiments. There's some measurement of that. But then you do you basically try to fit those parameters to what you see in rats. It is tricky to come up with parameters. There's not necessarily.
But is there any case where you could say this model is wrong?
Wrong. I mean, it's simplistic in a way. But it does capture the kind of dynamics that you measure experimentally in the cells. Yeah. So with this kind of description, we predicted some other types of some stable attractors that you only see in stochastic descriptions of this kind of system. And particularly in this system, we found that just the stochasticity that you get from gene regulation, from the binding and unbinding from proteins in the DNA, can already can kick the system out of this stable point and into this excursion.
So is that a prediction of your theory, of your model, or something that, again, you postulate the general model?
So what we do get from this kind of prediction is that we can actually tune noise from one source or another, right? Instead of like in this kind of description, you would put like just like a noise term over here, which is a very you don't really know where the noise is coming from. But with this kind of description, you can kind of tune on and off the noise from different sources, right? And the kind of prediction that we got is that just the noise from the binding and unbinding to the DNA, which usually people don't really consider, that can by itself create these entrances into competence. So this is another part of the network that's interesting to analyze, and I'm not gonna. I want to get to the antibiotics part, so I'm not gonna go into this in a lot of detail. But there's work done on this, and so this is the part that directly responds to the stress signals, and it has this cascade of phosphorylations between these proteins here, which is very heavily regulated, and there's lots of feedbacks. And what turns out is that this acts as kind of like a clock, where this protein here that regulates the entrance and phosphorylation, it has this like these peaks that are kind of correlated with the cell cycle, and the way it works is that you need high levels of this protein to make any of these decisions about the fate. So the cell creates these windows of opportunity for these stochastic transitions into the different phenotypes. So it times the cell decision. And if you go and look at the network as a whole, then you can try to divide it into these different modules that are responsible for many different things. So there's this two that we looked at. This one controls the entrance into competence directly. This is one that times all the cell decisions and then eventually controls the entrance into sporulation. And then there's these other parts of the network that they broadcast signals to the outside, and they send signals from other cells, so they coordinate the cell decisions at the population level. And then there's some other internal controls of the cell that basically control the commitment to each of these different phenotypes. So because the cell needs to choose one or the other. So this is once it commits to one, it just shuts down the other options. So before we go into the second part of the talk, like the take-home messages that I want to take to the second half now is that the gene expression is a very dynamical process, and variability in gene expression can lead the cell to different fates.
So this take-home message is something that comes out of the experiment, or something that comes out of the theory?
So far, it's only theory. Well, that was also experiment, right? So this coexistence of different phenotypes in an isogenic population in the same environment is something that you see experimentally, right? And then we made some modeling to try to explain what's the origins of that.
I understand that, but why need the modeling in order to take this take-home message?
I get so okay. So the diversity of phenotypes in a population sometimes they see experimentally, and there's also a bunch of other experiments that show the gene expression, especially in bacteria, is a very noisy. Is very noisy, right? So yeah, we have experiments that show that we have a lot of noise in expression, and you do see a lot of variation in phenotypes. So the theory part here was try to understand how the cell uses this noise to its advantage to basically regulate at a population level which cells go into one phenotype or another. So basically how the cell can actually use noise to at a population level have a certain division of labor where you have different phenotypes like doing different things for the benefit of the colony. Yeah, is that a...
The bigger part is model to explain the different the ratio of number of
So yeah.
And we were successful in explaining this?
Yeah. So it explains. Of course, like you need to fit some parameters, but yeah. You basically explain that. Yes. Okay. So now I'm going to talk about the antibiotic resistance thing. So first, as an introduction, so usually if you talk to biologists about antibiotic resistance, usually they usually think of it as like a steady state thing. So under a certain concentration of drug, are cells able to grow or not? So if you have cells and you explode and there's a certain concentration of antibiotic in the environment, do you see growth or not? But the picture is a bit more complicated than that, because usually this resistance mechanisms, this antibiotic resistance mechanism, they express proteins that are costly for the cell. So it carries a cost. Their expression carries a cost. So since usually the cells they're not constantly exposed to antibiotics, they usually repress the expression of these resistance genes in the absence of the drug. So what happens is that then when the drugs come into the picture, then the cell has a short window to act. So it needs to efficiently express these resistance genes to be able to resist the drug. So I'm going to show here that what happens like at the moment that the cells see the drug, that is very critical for the survival of the cell. So there's more than one. A cell can acquire some level of resistance to antibiotics through mutations here and there, but that's not what I'm talking about. What I'm talking about is, which is more clinically relevant actually, is this whole mechanisms of resistance that the cells can acquire. So it's genes that get rid of the drug one way or another. And they're usually present in these mobile genetic elements that they can be transferred from between different cells, and sometimes even from species to species. So they're pretty nice systems to study because they're usually only a few different genes, and they're disconnected from the rest of the cell metabolism, and they have their own evolutionary history basically. And especially nowadays, in the past few decades that we've been using antibiotics, that they usually have their origin in soil bacteria, and now they're actually, in the past decades, transitioning into pathogens. Right? So it's like an evolution that's happening within our lifetimes, that these resistance mechanisms are adapting from environmental bacteria to pathogens. and they're exposed to very different selective pressures, right? So the way that the cell sees antibiotics in the environment is very different from how they see it in the clinic. So it's very interesting to analyze. So here, for instance, is a map of the world where each country is colored by the percentage of environmental E. coli that shows some sort of resistance to beta-lactams, which is like an important class of antibiotics. And you see that some countries is basically all of them. Okay, so for the rest of the talk, I'm going to be talking about this one particular resistance mechanism. It is maybe the most studied resistance mechanism in the most studied bacteria there is. So it's an E. coli, and it confers resistance to tetracycline. And it's this classical example of drug-induced antibiotic response. So it's only these two genes, and it's present in one of these mobile elements, And this repressor here is very typical of species that live in variable environments. They need to change with changing environment. So this is basically how it works. So tetracycline is an antibiotic, right? And it diffuses through the cell membrane, and it binds the ribosomes and prevent it from making protein. So it's an inhibitor of translation. So here's the resistance mechanism in the absence of drugs. So you have this repressor being made, and it binds to the DNA here and prevents transcription of both the stat-A, the efflux pump, and of itself. So then when the drug comes into the cell, it binds to this repressor, and it loses the capacity to bind DNA, so then it releases the expression of both itself and the efflux pump. And now the cell makes this efflux pump that pumps the drug out of the cell. But this comes at a cost, because in the process of expelling the drug from the cell, it also messes up the membrane potential here, which cell needs for a lot of different functions. So if the cell makes too much of this efflux pump, then that is also toxic for the cell. So these are the two characteristics that we're like really interested in this system. So it's very strongly repressed, and it poses a trade-off in the expression of the resistance gene. So the cell wants to make it to resist the drug, but also doesn't want to make too much of it. So there's an optimum level that the cell will hit. Yeah. Yeah.
You want to kill the cell?
Me?
No, the antibiotic wants to kill the bacteria, right?
That's right.
So the idea is to kill the cell, or not?
The idea is to kill the cell, yes.
Okay, good. And then you're saying that if you put too much antibiotic...
But the cell doesn't want to die. So the cell wants to induce this response efficiently in a way that it won't die either from the drug or from making too much of this efflux pump. Yeah? Okay. I would love to, yes. That's very, very good to get grants. Okay. So then what happens when you put this in cells that carry this resistance mechanism? Like what happens when you expose them to a certain concentration of the drug? So here we're comparing cells. This one colored in black, they don't have the resistance mechanism, and these ones do. And this is a growth curve, which is basically if you put cells in culture medium, they're going to grow exponentially until at some point they saturate, right? So this is basically the measure of the population. So the log of the population. So exponential growth here is going to look like a line, and then eventually they're going to saturate. So this is what you see in the absence of drug, and you see that the cells that carry the resistance mechanism and the ones that don't, they basically grow at more or less similar rates. So here's what happens when you, at a certain point during growth, you add a certain concentration of drug. So these are these are pretty low concentrations. And you see that the resistant cells, they don't even see it. But the sensitive cells, they start growing slower, right? Or if you add enough of it, then they don't grow anymore at all. And the resistant cells, they're doing just fine. But then if you increase the concentration of drug, then for the resistant cells, actually you see something different. So what you see, the pattern that you see is that in the steady state, a while after you added the drug, they actually grow at pretty similar rates as before you added the drug. But you start seeing this delay here, right? Which looks kind of odd. So we looked at the microscope to see what that's all about, and actually what happens is that right as you add the drug, you kill most of the cells, and only a fraction of them actually survive. So like see, this is like a moderate concentration of drug at the time that you add the drug, and then later you see that most of the cells are growing, but there's some that are not. This is like a higher, yes? I'm sorry? Oh, this year? Yeah, so, yes. You add them at a certain point, but you add different concentrations. So higher and higher here. Yeah? Okay. Yeah, so this is a higher concentration, and you see that you kill most of the cells, and only a few of them are growing. So this is how it looks in a movie. Like, look, there's a bunch of them are dying, but then there's a few of them that are able to survive. So now we kind of use this to kind of have like roughly calculate like two measures of performance here. We look at when looking at these growth curves. So we look at this steady state growth here, and more or less of like a measure of how well the cell does in the steady state, like after everything is stabilized. And this delay here is more or less of a measure of like of what happens right as the drug, how well they do upon impact. So the longer this delays, the more cells were killed when the drug was added. Okay, what determines cell survival? So ideally the cells would just jump expression to this optimum that's basically the optimum on this trade-off of expressing the efflux pump, right? So what does it do? Does it just make a lot of efflux pump right away and risk having like this large overshoot which is toxic, or does it take it easy and go slowly, but then they risk getting killed by the drug? So do they underdamp, overdamp the system? So we wanted to measure this. So the answer to this is in the regulation, right? So we want to look at what the repressor was doing. So first thing that we did here was we made this synthetic circuit here where we put this the repressor under an inducible promoter. So basically you can add a molecule there and induce the repressor directly. So basically you bypass the feedback loop there, the activation. So this is what happens in the absence of the drug, right? So the more of this inducer you add, the more of the repressor you're going to have, and you're going to have less and less of the efflux pump. So then we grew cells in these different concentrations of the inducer. So this was their state as they were exposed to the drug. And first we're going to look at this one here where you didn't induce the repressor at all. So it's the cell that had the most efflux pump to begin with. And it actually does very poorly. So So this year's growth without the drug, and then you see that it has like many hours of delay here. So actually those cells didn't do that well at all. While these cells here, they have a certain level of repressor, even though they have less efflux pump at the time they had the drug, they actually did much better. So what this means is that the native promoter of the efflux pump, if unregulated, is making enough of the efflux pump to kill the cell. So it's very important to regulate it. So because if left by its own devices, it can kill the cell. It's very strong. So okay, so what have we learned so far? So the response requires that the cell induces this efflux pump promptly, but it needs to keep this expression at safe levels. And expression of the repressor together with the efflux pump actually helps. So how do you optimize this response for the transient? So now we're going to look at the simplest model that I could think of at the time here. So we wrote this equation here to basically keep track of the intracellular concentration of the drug. So it has a term here that is basically a diffusion of the drug inside the cell. There's another term here, which is the expulsion of the drug from inside the cell by the efflux pump. And there's another term here, which is the basically dilution of the cell's components because of cell growth, right? So it's basically like the degradation term. And then we consider this a binding equilibrium between the repressor and the drug. Okay, so it's just the one equation that keeps track of the concentration of the drug inside the cell. Sorry? So this is the concentration of efflux pump. Okay. So then we're going to do something really simple here for the regulation of the genes. We're going to consider that they are repressed in the absence of drug, and then once you add drug outside the cell here and it starts diffusing inside the cell, we're going to consider that they are expressed at fixed rates, and these are going to be the two parameters that we're going to play with: the rate of expression of the repressor and the rate of expression of the efflux pump. And then we're going to consider that there's a hard limit here on the regulation of TetR. So basically, once the concentration of free repressor crosses a certain threshold, you shut down expression of the efflux pump. And then we're going to consider that these two things can kill the cell, like a high drug concentration or a high efflux pump concentration. Okay, so then if you simulate this, this is like a typical time course of what happens. So this is the drug here. So once you add the drug, so then drug starts accumulating inside the cell, and then you have a synthesis of the efflux pump and the repressor. And then efflux pump starts accumulating. At some point, you have enough of it that you start reversing the influx of drug inside the cell. But then the efflux, and at this point, if that happened below the limit here, the cell avoided the first death from drug. But then the efflux pump concentration is still growing. And then at some point, the drug concentration goes down, and then you start freeing up repressor, and then and that shuts down the expression of the efflux pump. So then, if that happens below the limit here, then the cell avoided the second death from making too much of the efflux pump. So now we can simulate this for different values of this synthesis rates, and see how much drug basically the cell can resist. So this is in the absence of any repressor. So the more efflux pump the cell makes, it resists more, up to a certain point where just this constant production here is going to cross the toxic limit of the efflux pump, and then the cell just dies from that. But then as you increase this synthesis rate of the repressor, then the cell is able to also increase the synthesis rate of the efflux pump, because now we gain this ability to shut it off when it doesn't need it anymore. Right? And so then in this way, the more repressor you make, the stronger the synthesis of the efflux pump the cell can have. And then what's interesting here is that if you look around one of these optimal points here, like even slight variations in the rate of expression of the repressor can already lead the cell to different fates. So you can have a coexistence here, like with a small window of synthesis rate of the repressor. You can have cells dying from too much drug, dying from too much efflux pump, or recovering. So in the simulation, it looks like this. So this cell here, that's you can have the drug crossing the limit, and then the cell dies from that. Or if you can avoid that first death, it can die from the second death here, from having too much of the efflux pump. Or it can avoid both of them and survive.
Sorry,
Yeah.
this plot.
Mm-hmm.
How, can you actually see the time delay? You showed a plot where it was a time delay.
Yeah.
In giving antibiotics and the cells growing again. Can you see from these plots where the time delay comes from?
So the time delay, we looked at it in the microscope, and the time delay comes from most of the cells dying, and only a few of them surviving. So now you have all this background of arrested cells, and only a few of them are growing. So then you only see growth again once the surviving fraction grows above this background of arrested cells.
That time delay
So,
is not related to this time.
so that time delay would basically mean would relate to the proportion of cells that end up dying, basically. And we're gonna say about that in a bit.
Because that was hours, and this is minutes. Time delay was usually hours?
Well, yes. So this, I mean, this depends on the parameters that we use, right? This is a pretty simplistic model, but I can tell you that this happens like in the first 4 hours. Yeah, but I'll show something. Okay, so here's this phase diagram where I have the drug and efflux pump concentration that shows the zones of death here that the cell needs to avoid. And so this is a plot of the response, and then you see that if you increase the synthesis rate of the repressor, you increase the responsiveness here. It's quicker to avoid both this death and this death, and stay like within this survival area here. So yeah, so the idea is that the cell first wants to have low concentration of repressor, so it will be sensitive to the drug coming in, but later it wants to have more repressor to be able to shut off expression of the efflux pump when it doesn't need it anymore. So this is somewhat analogous to this notion that we know from control theory, that if you want your system to be responsive, you increase the gain of the negative feedback. Yeah, so here it just shows that the more repressed, the fastest the cell can make a repressor, the better it approaches the resistant level of a step response. Okay, but then yeah, so do you actually see this in nature? Do you actually see this three fates? So we looked at this. So we made this little device to try to see like what decides the fates of the single cells. So this is a microfluidic device. So it is this microfabrication technique where you can make really tiny channels that carry fluid, and our design here has a larger feeding channel here, and many of these very narrow side channels that have a dead end here, and they're the width of a single cell. So you can put cells inside them and spin them in the centrifuge and get these cells stuck at the end here. And then they grow and divide, and then the daughter cells just get washed away. But then the cell at the top there, you can keep looking at it for basically as long as you want. And we made it so you can switch the medium. So basically you can grow them on just fresh medium for a while and then switch to medium with drug and see what happens. So we put these fluorescent reporters on the two genes, on the efflux pump and the repressor. And then the idea is that at some point you add a pretty high concentration of drug, like about at the limit of what the cells can survive. And then look at gene expression and see which cells survive and which ones don't. Okay, so this is it here. So, okay, I'll start. So at time 0 is when we add the drug. And green is the repressor, and red is the efflux pump. So you see that, like, okay, now I added the drug, and then the cells, first they turn a little green, and then you see all kinds of things happening, right? So these cells are all the same. They are all from the same from the same colony. They all have the same resistance mechanism. But you see a lot of variation at the single cell level. So basically here you see three patterns. You see that some cells that they die very dim. You see the cells that recover that they're the yellow ones that make both of the proteins. And then you see these other cells that die very red. They make too much of the efflux pump. So basically, these are the three fates that you see. So this is recovery. These are cells that they grow a little bit initially, and then they turn really red and die. And then there's these cells that just basically stop growing right away without expressing much of either protein. So basically this is their accumulated growth colored by cell fate. So they're the cells that keep growing, the cells that grow a little bit initially, like, and you can't really distinguish them at this point, but then they stop growing, and then they're the cells that die right away. And this is what you see from expression. So basically the cells that survive are the cells that were able to make the repressor of the efflux pump the fastest. And they make the efflux pump quickly initially here, but then they stop. But then when they reach the level that they want, then they're able to stop making the efflux pump. And the cells that had intermediate levels of synthesis of the repressor are the cells that they were able to make the efflux pump quickly in the beginning, but then they never stopped, and then they make too much of it. And they die. Those are the ones that die very red from making too much of the efflux pump. And then there's the cells that just don't really make much of anything. And presumably they died from the drug initially before they could synthesize any of the proteins. Which basically like are these the three fates there that we saw from the model. And here is in this diagram here of repressor and efflux pump, they plotted at different time points here. And you can see already within the first hour, you can already see that you can already tell what's going to happen to the cells by their TetR expression. So actually, if you use this measure of like if we use the information gain approach that basically calculates the best split of the population here. to try to see like what's what of all these measurements that we make, what can best predict the future of the cell. And you can see that how quickly they can make the repressor is the best predictor of the cell fate. And you can already tell that in the first hour, and it's better than growth. So you can actually, by how much repressor they're making, you can tell what's going to happen to the cell while the cells are still growing, basically. Yeah, so the repressor TetR is the best predictor of cell. Okay, so basically, what we learned here is that proper induction of these resistant genes is essential for the cell survival. And actually, the kind of a bit of a counterintuitive result here is that the faster that the cell make the repressor, it optimizes the dynamic of induction. So it's good for the cell to make a lot of repressor right away. And then we see that the variation in the expression level of the repressor can lead to different cell fates. So I want to just like put this picture here of how dynamical this process is. So usually the way you think about this is that this repression curves here. So basically, in the absence of the drug, the more repressor you have, the less efflux pump you have, right? So in the absence of the drug, then the native circuit is going to sit at some point along this curve here. And as you add the drug, then the drug takes the repressor away. So basically you shift this curve, and then the native circuit is going to sit like somewhere along this other curve here. And we're putting this picture here of how important the dynamics of the whole thing is. So like now we have like an initial steady state and a final steady state, which are these two here. But it kind of matters how you get there. So these are the average trajectories that we saw in the microfluidic experiment. And you see that if the cell doesn't follow the right trajectory to the second steady state, it actually can get detoured and die. Okay, so how am I doing on time? Are we in a hurry? Or? Few more minutes? Okay. All right. So now this is something we kind of just started doing recently. So I'm going to be quick about it. So we want to see like how how does the cell actually do that? So how does the cell design the regulation to optimize the dynamics of the response? So this is actually what you see in the TET operon in nature. So this is how it looks in the DNA. So it's these two genes that they're expressed divergently. So this gene goes in that direction, this gene goes in this direction, and in between them, there's 80 base pairs. There's only 80 letters that pack a whole ton of regulation. So each of and it's all overlapping, and each of these rectangles here are spots where the polymerase can bind and initiate expression of there's one of these for the efflux pump, and there's two sites where it can initiate expression for the repressor, and then there's two binding sites for the repressor here. So how does the cell arrive to this design? Is this actually optimal? So what kind of disposition of these regulatory elements actually achieve the regulation that optimize this response? So I'm just going to be really quick here, but basically what we try to do is run an optimization problem on this. So basically we would we would start with a configuration of these regulatory elements, of promoters and binding sites for the repressor, and treat this binding affinities of the polymerase to the promoters and of the repressor to these binding sites as parameters that we're gonna try to optimize. And then we design a cost function that has what we think is the bare minimum that you need, which is basically putting a penalty for expressing the efflux pump above a limit, putting another penalty for having drug go above a limit, and also putting a penalty on protein expression. So put a cost on how much proteins they use in the response. So then now we have this system of three equations here. That's the one that we saw before that keeps track of the drug inside the cell, and then these two other ones that describe the concentration of the repressor and the efflux pump, and their synthesis here is a regulatory function that we get according to the disposition of regulatory elements and these parameters here. Okay, so then we start with a configuration. We run a simulation. We calculate the cost. Then we optimize the parameters. And then we can use that as a starting point and do this again for a higher concentration of drug. So then we keep increasing the drug concentration and running this optimization in a way that, given an initial configuration of these regulatory elements, we have the optimal cost as a function of drug concentration, and basically we can see what kind of regulatory function the cell ends up with. So then we made all the different combinations of these regulatory elements. And we can compare them against each other to see how good they are. So this is the cost and drug concentration. This is what you get for only one regulatory element. So the cost goes up very quickly initially already. With two regulatory elements, like this is the best you can do. Also, it's very bad. And now with three regulatory elements, now you start. Now you see. Now you have regulation, right? So now you have, and this is the best configuration for three regulatory elements, which is one unrepressed weak promoter for the repressor, and then a stronger promoter for the efflux pump that has a binding site in front of it. So you see that the expression of the repressor doesn't change; it is not regulated, but now the expression of the efflux pump is. So now if you add another regulatory element, so now the best solution that you get is if you add another binding site of the promoter, of the repressor here. So now you have two binding sites in front of the promoter for the efflux pump, so you get like a sharper transition here. And you have one in front of the repressor, so now the repressor also self-regulates. And if you add another one, then you don't really gain much from that, but then you get this solution here, which is basically you end up with like more reasonable parameters for the binding affinities here, and it turns out that this is exactly the disposition of the regulatory elements that you find in this TET operon. So yeah, basically that's the best solution that we also found. And it has this characteristic. So it has a very strong expression of the efflux pump with a very sharp transition. So the cell is able to induce it very quickly once the drug is present. And in the absence of drug, you need very little repressor to repress everything. So it's very sensitive to the presence of drug. And you get really fast activation of the efflux pump, and then you also have very fast repression of the efflux pump once the cell starts to expel the drug from its interior. Okay, so here we see how complex regulation emerges to control the dynamics of the cell response. We see that the TET operon, actually the disposition of the regulatory elements that it has there actually optimizes this response to a sharp increase of drug concentration. And this is kind of what we want to do, is to kind of start like decoding the information about the patterns of expression of the genes that's also written in the DNA, right? So I'm just going to leave with this picture here that we would like to be able to look at the genotype, and then from there, like, make inferences about the phenotype of the cell, like what kind of pattern of gene expression that you're going to get. And then from the phenotype, we would like to assign a fitness to that. So under a certain environment, we can kind of start thinking about predicting of like a certain, how if you expose a certain genotype to a certain environment that has like this type of fitness landscape, we can try to start thinking about predicting what is the optimal genotype to have in that environment, and like what kind of trajectory, evolutionary trajectory can it has in that direction. Okay, so the middle part of the talk was done with Roy Kishoni and Adam Palmer at the Harvard Medical School, and then at the Technion. The initial part of Bacillus was I'll mention my advisor Peter and Eschel. This is at UCSD, and more recently working with Catherine and Lev at Dartmouth and at the BCI Biocircuits Institute at UCSD too. And then thanks. Sorry, I went a little over. Mm-hmm.
Just a very naive question about the first, before you move to the very last topic. Until then, do you do quantitative comparisons, or are you mostly interested in the general profile of the curves?
Okay, so we try to be as quantitative as we can, but then, say, for instance, one difficulty that we had is, so say, for instance, we were interested in how this variation in transcription level correlates to the cell fate, right? And so our setup is, so one limitation of the setup that we have is that we have the resistance mechanism itself was in the chromosome of the cell, and the fluorescent reporters that we use to read them is this fluorescent proteins that are under the same promoter as the resistance genes, but they're in a plasmid. So basically the variation that you see from the fluorescence is not exactly the variation that we see from that you would see in the resistance genes, for instance. So
maybe let me ask differently. So is it fair to say that you knew more or less the answer already? I mean, you knew the cartoons, you knew already, right? From the pictures when you start explaining.
That's how I put the talk together. Yes.
But is it true or not? Maybe that's what. Maybe this is what I don't know. So my feeling was that you knew already the cartoons, right? These either they die like this, or they die like that, or they die like that.
We did them at the same time, but yeah.
So I could understand that from the experiment you could also check that, right? So the experiment is awesome. You see that they die in different ways, and from there you could confirm the cartoons.
Yes.
What I'm just not understanding is why the differential equations, because since it's a phenomenological toy model anyway, my feeling is that you would at best compare that you get roughly the curves. But then what's the advantage in having these models that you don't believe that quantitatively they should work, right? I mean, they should just give roughly the mechanisms. Or do you really think that if you add enough microscopes, you should be able to compare various derivatives of this curve?
Yeah, I think so.
Yes?
So the idea of the model is to try to write the simplest model that you can that would capture this kind of behavior, right?
Yeah, I just don't. But how could you control the approximation of the model? How could you say, in what sense is it a controlled approximation? Say in physics, we have, say, expansion at high temperature. And we say this is first term, second term, third term. The more terms I put, the better it works.
Right.
Well, what's the controlling parameter here? How do I know that I get the first approximation right and then a little bit better?
Well, so these models are very minimalistic, right? Because we're trying to basically what we're trying to see is like what kind of interactions do you have to include in the model? Like what's the minimal model that's going to explain the behavior, right? And the reason why we picked this particular system is because it's very well studied. So actually, there's not that many parameters to vary, and a lot of them are actually measured. So we're not fitting too much here.
Or maybe let me ask just one last question. So from experiment and from your intuition, you already had this picture of these rough curves of what could happen. Is it fair to say?
Well, we got our intuition from experiments initially, but yes.
And then you developed the mathematical model, and now you could compute many things in the mathematical model, right?
Mm-hmm.
Did you unveil in this way any mechanism or any surprise, or did you learn anything that the experiment and your biology intuition did not tell you? Was there some novelty that the numerics gave you?
I would say that the surprising thing that we got here is that that the cell has this need to express the repressor very quickly, right? And that's what the cell that's what allows the cells to survive is actually this strong expression of the repressor of the efflux pump, which is kind of counterintuitive, right?
No, but that you could see from the experiment as well, right?
Yes.
I meant from the differential equations, from the theoretical part. Was there something that you learned that you did not know either from the experiment or from biology?
Well,
did you learn something from the quantitative analysis?
I mean, I guess we learned that if we include this very minimal approach of basically just including just diffusion and efflux of the drug, that alone already that's enough. That there's not and then we can we can be quantitative about like what one thing that we did get from the model, for instance, like in the mid part of the talk, is that we did see like from this phase diagram, basically we see this like coexistence of the of these different cell fates around this optimal point of regulation. So say if you have an optimal regulation that is exposed to basically the highest concentration of drug that it can survive, right? Then one thing that we did get from the model is that because the drug inside the cell we can only get from the model, right? That we can measure really. We can measure the expression with the fluorescent proteins, but we can't really see the drug. But one thing that we do learn, for instance, is that in an optimally regulated system exposed to a high concentration of drug, that the cell really like almost dies from both of these deaths. And little variation that actually you can see the coexistence of these fates, like with a short window of variation in expression, for instance. So that's something that's something they get in the model. Yes.
So I was surprised. I mean, why is this mechanism so well adapted? So are there other mechanisms for antibiotic resistance? So I mean, antibiotic resistance is a big deal, and apparently bugs are increasing their ability to respond to antibiotics. But in this particular case, what you show is that it has already saturated, so this particular bug does not have any potential for improving antibiotic resistance, right? So, so the general question is, how relevant is this to understand antibiotic resistance in general?
So most of these antibiotic resistance mechanisms, they are regulated in one form or another, right? But they depend a lot on the mechanism of action of the drug. So say, for instance, if the cell wants to use efflux pump to pump drug out of the cell, there needs to be a drug that doesn't get very readily imported into the cell. So say, for instance, so in this case, this works because tetracycline takes a long time to diffuse into the cell. Like it's in the order of like 40 minutes, the half equilibration time. So it doesn't flood the cell. So then it can actually use an efflux pump to pump it out, for instance. But then another thing that is typical of this is that this efflux pump is pretty toxic for the cell, right? So depending on the mechanism of action of the drug and the mechanism of action of the resistance mechanism itself, then basically the constraints or the problem can be pretty different, right? Which would, and then presumably the cell would arrive in different kinds of regulatory solutions for those problems, right? But then there's not that many combinations. So then these are things that this kind of designs are things that you do see repeatedly. Like if you look at of different mechanisms that act against different drugs. So they are so it's general in a way, if that's a.
So and why should it be optimized for this particular response to a sharp increase of the drug? I mean, you could think of solving the problem of finding what is the most dangerous thing that you could do to this bug. I mean, what is the protocol that?
So that's kind of the direction that I actually want to pursue, which is to look at what are the optimal solutions for different environments, for instance. So say when usually these resistance mechanisms they originate in soil, and the bacteria have been making antibiotics and resistant to them for like 1 billion years in the soil. Right? But it's not thought to. The antibiotics are not thought to reach very high concentrations in the soil. They're not like a predatorial thing where the cells make antibiotics to kill everyone around. It's more like a warning signal. Right? So the regimes of drug that a cell would see in the soil is presumably very different from what you would see in the clinic, where you just give like a ton of antibiotic all at once to a patient, right? So one thing that I do want to look at, which I think is pretty interesting, is like it's like what happens in this adaptation then from resistance mechanisms going from soil to a clinical setting, for instance. So what do they have to change in the regulation to adapt to a new environment?
Yeah, so maybe very related to that question, I remember some work from Paul Francois, maybe you've seen it, where he evolves...
in Canada, right?
Yeah,
Uh-huh.
Genetic networks. So, I mean, do you see this same type of genetic circuit in response to some other antibiotics? And have you thought about how it might have evolved, you know, from the cost functions that you set?
So say, for instance, one thing that you see a lot in this resistance mechanism is this pattern of like divergent expression of the resistance genes with a repressor, that the repressor also regulates itself. So that you see very often. Like the exact disposition of the regulatory elements, I don't know like a whole ton of examples, but one thing that I would like to do, for instance, is like I don't know, get like the whole E. coli genome and look at all the intergenic regions and find like binding sites and operators and like that to like, you know, like have like a library of like all these different regulation and see try to correlate that with the kind of response that they're involved with, for instance.
Maybe, I mean, do you think you could apply this to antibiotic therapy? Is there a way that you say an human is infected, let's put his bacteria in the regime where they become arrested or something?
Well, so for instance, one thing, two things that could be explored are if you know, go for treatments where you can hit the bacteria harder, like so say, I don't know, maybe that can educate the clinical. So instead of taking a pill where the drug takes forever to get to the site of action, you can just get IV, for instance. Or you can think about treatments that, together with antibiotic, you can also take something that disrupts the regulation that induces the response, for instance. So in that sense, yes.
Any other questions?
Hi.
Hey.
What kind of kinetic information did you need to develop the model? Kinetic information about the proteins, how fast they react, you know, the Hill coefficient. Did you have to measure something about the proteins before or after the model?
Okay. So
and what and how?
So good question. So some of it we got, some of it we measured ourselves. So say, for instance, what's the toxic level of expression of the efflux pump? So we put it under like an inducible promoter, and we made more and more and more of it until like we saw like how much was toxic. Or like same thing with the drug, right? We can like expose to a drug gradient and see like how much of it is toxic. So we measured like some of that. And then other things you get from the literature, for instance. So this efflux pump is pretty well studied. So people have people have rough calculations on like what's the efflux rate that this efflux pump achieves, or like how well it binds to the drug, or people know the binding equilibrium, the equilibrium rate of the binding of the repressor to the drug and of the repressor to the DNA. Yeah, so a lot of this kind of information like is there in the literature. It's not necessarily usually the case. Also, they have information on the diffusion rate of the drug across the membrane. So there is so for this particular system that's very well studied, you do know a lot of these numbers.
Is there some data bank with this information?
I'm sorry?
Is there some data bank with this information?
A big database with like this biophysical parameters. I am not sure. I got it in the literature. There's a lot of this for this system. There's a lot of papers from the 80s that measured all of this. Yeah.
Okay, so let's thank Daniel again.
Thank you.
Thank you.
Oi.
Pode ser?
Claro.
Você coloca o antibiótico, as
Uhum.
células morrem
Isso.
e sobram algumas células que vão dar um boom.
Isso.
Essas células que sobram,
você analisou